So we just finished up talking about fatty acids. Let's cover the other three types of lipid that we want to be familiar with in this class. Up next, triacylglycerol. Why is this lipid called triacylglycerol? Well, the backbone, if you will, of this lipid is glycerol. So 3 carbons, each with a hydroxyl on it. We say triacylglycerol because when we add in these lipid tails, we're adding them at each of those hydroxyl groups. So we are acylating the glycerol 3 times. Triacylglycerol. And so what a triacylglycerol molecule looks like is here on the right. We have this glycerol backbone up top, followed by this ester functional group and the rest of the lipid tail. Most physiological lipids, like the lipids being stored inside of our body, appear in this form. Usually, each of these 3 lipids is different from one another. Some are saturated, some are unsaturated. Triacylglycerol is the only type of lipid that is not amphiphilic. You might be thinking that this ester functional group has some polarity. You would be right. We could draw dipoles, but it is insufficient polarity to make this molecule amphiphilic. As mentioned, triglycerol is super common in our bodies because it is how we store our energy in fat cells. It is highly reduced, right? If you take a look at this long carbon chain, we're not seeing many oxygens, we're seeing a lot of hydrogens, so it's very highly reduced. As we'll learn later in metabolism, things that start off super reduced will give off a lot of energy in our metabolism as they are oxidized. And these are really packing a big punch. They provide 6 times more energy per unit mass than a carbohydrate that we eat, which is pretty cool. The next type of lipid that we should be familiar with are the phospholipids. These are important because they are the major component of all biological membranes, our lipid bilayers. They have a slightly different structure, and it's actually a bit more clear on the next slide. So let's jump to that slide just real quick. Okay, I like this slide better because it shows you the correct arrangement of this type of lipid. So the phospholipid has a phosphate in this kind of head region on the lipid that's pointing up. Okay? It is coming off of one of the 3 carbons on this glycerol backbone. Now, since one of the glycerol carbons has a phosphate, that leaves the other 2 glycerol carbons connected to these lipid tails. So that is the type of structure we expect to see from a phospholipid. Now, because we have that phosphate group coming off the top, it has a negative charge, we definitely consider these molecules to be amphiphilic. There is a very charged region, which is certainly polar, and then of course the lipid tails are nonpolar. So therefore, yes, amphiphilic. What else should we know about our phospholipids? Well, I already mentioned that we have these 2 lipid tails. A lot of times they're kind of simplified as R1 and R2, but we should know there's like a long hydrocarbon chain there. Carbon number 1, so this is carbon number 1, oftentimes is a fully saturated lipid tail. Carbon number 2 is oftentimes unsaturated. And then carbon 3, of course, is attached to that phosphate. Now, in addition to having that phosphate in the head group, there is what we call just like the X We don't want to use R because that's a hydrocarbon chain, right? So this X can be variable. It can be something like an amino acid. It can be something like a sugar molecule. It can really vary depending on the lipid. So you may have heard of like phosphatidylserine, phosphatidylcholine. The serine, the choline, those are what is attached to the phosphate at this X location. Last but not least, let's talk about the last class of lipid before moving on to more of the structure function understanding of lipids. The last class is steroids. They have a very different structure than the other three classes that we just talked about because they have this 4 fused ring structure, where we have 3 of the rings have 6 members and the last ring is 5 members. Now, steroids are made from fatty acids, so of course we consider them a type of lipid. It's just their synthesis is even more complex. Now, steroids are very important for our lipid bilayer, okay? They make up 30 to 40% of our membranes, specifically cholesterol, right? Cholesterol is an incredibly important aspect of our bilayers. We do consider steroids to be amphiphilic because, for example, cholesterol has a hydroxyl group sticking up. In the space-filling version here on the right, that hydroxyl is right at the top in red. Now, this hydroxyl is sufficiently polar for us to consider steroids amphiphilic. An ester group, not sufficiently polar, but this hydroxyl group is sufficiently polar. The steroid cholesterol is the precursor of many steroid hormones as well, so incredibly important for our biology, right? Think estrogen, think testosterone, think all of their derivatives. Super important to our biology. Next up, we're going to talk more structure function. Why do lipids really matter, and how can we start to understand them?